So I think it was about 6 weeks ago, Dave Yallerly sent us an email saying, we need your seminar titles yesterday. And I made a snap decision to present some work that isn't perfectly polished or published yet. And part of my reasoning for deciding to present work that's in progress is because I'm a new CI in our new centre. So I wanted my talk today to convey the new directions my research is taking. And the other reason is that this work is at a stage where there's some exciting patterns emerging, but it's not too late to incorporate additional elements to the research I'm presenting. So do contact me with ideas and suggestions. So I was quite envious of Carissa's slide earlier, where she had a much more fancier high-tech schematic of these processes than I have here. But I'd like to use my low-tech version to reiterate the point that coral reef ecosystems don't exist in isolation. So this is a seascape of coastal landscapes moving out to reefs. We know mangroves help prevent erosion, they absorb nutrients and also freshwater discharge from terrestrial environments. Seagrass beds lying in between mangroves and reefs can help absorb sediment. They absorb nutrients and bind sediments. And coral reefs also have an important role in exchanging nutrients and other processes between ecosystems. So the one I'll highlight today is that reefs actually have an important role in exporting organic matter and nutrients toward the pelagic food web. They do this by releasing propagules. Those eggs that they produce during mass spawning events get exported into the pelagic environment, and also because reef-building corals actually excrete a lot of the carbon that they get through their symbiont photosynthesis directly into the water column. So in addition to things like fisheries goods and marine natural products that reefs provide to societies, there's also a range of other ecosystem services. So I've highlighted some here that we haven't really spoken about in a great deal during the symposium so far. They're things like biogeochemical processes. Reefs have a role in nitrogen fixation. They have a role in calcium and carbonate budgets on a kind of global scale. And they're also, the corals within reefs have a role in waste assimilation. So the symbionts that live within coral tissue can actually assimilate nitrogenous products that are the waste products that other organisms release into the environment, which is quite amazing. As I mentioned on the previous slide, they also have this role in export of organic production to pelagic environments. So what you're looking at here is a Google Earth image of some reefs on the outer barrier off Lizard Island, and also the Swains Reefs down in the southern part of the Great Barrier Reef. And I'm using examples from these locations to highlight the fact that coral community composition, so the species composition of reefs, varies systematically along these kinds of gradients. So this is part of a much larger study led by Ashley Frisch, also with 2 PhD students from our centre, Jordan Casey and Justin Rizzari. So it's a much broader study, not looking just at coral community composition, but also at trophic links, so counting apex predators, surgeonfishes in different reefs along these gradients. So in each of these 2 regions, we'd visited 2 or 3 pink zones, so they're the no entry zones. Also the green have the protected no take zones, and the blue general use or habitat protection zones. So visiting different management zones within these regions, there were 4 sites per reef, and then we had 4, at least for the benthic data, 4 50-metre point intercept transects per site. I'm not going to go into these data in a lot of detail. I just want to illustrate the point that we see changes in community composition between these 2 regions. So this is just a multi-dimensional scaling representing the coral community at each location as a single point on this graph. So we have the Swains Reefs clustering in one section up the top, and the Lizard Island Reefs clustering roughly together, or at least distinct from the Swains Reef in the middle. So showing that, yes, the coral composition at these locations is varying between these regions. And the change is dominated generally by an increase in those more heterotrophic, fleshy, mucid species in the Swains compared to Lizard Island. Also, in kind of similar to the results that Gary and others have showed us this morning, showing no real differentiation in coral cover between management zones, there's no clear signal of a change in coral species composition between pink, green and blue zones at these scales, which reflects the fact, I think, that coral community are structured by processes other than ones that are controlled by management zones. So we do see these differences in coral community composition across gradients. And we don't have any systematic study of whether these changes in species composition lead to changes in the export of organic matter from reefs, for example. So these are some data from the literature, a compilation of data kind of showing growth rates of different branching versus massive colonies, and also things like reef productivity of communities that are dominated by different coral species. So we see for productivity in the center, acropora communities are having higher productivity than mixed coral assemblages. For calcification, some indication that these acropora communities might have higher calcification, but not different, at least at this data compilation level, from the mixed communities. So we know coral communities are changing. There's some evidence that their overall reef productivity or the amount of carbon they're exporting to pelagic environments might change with community composition, which is what's motivated the work that I'm talking to you about today. So the title of this talk was Effects of Ocean Warming on the Production of Coral Communities. So hopefully the context I've given you so far has introduced that we might expect to see changes in this production, productivity, calcification of reefs, given changes in community structure. If we want to go to answer this question in a systematic way, we need to obviously account for interspecies or among species variability in these productivity levels. We also need to incorporate spatial and temporal variation in the environment. And then we also need a strategy of scaling up from processes happening at polyp level or individual colony level to ecosystem scales. So today I'll be focusing mainly on these first 2 points, just in the sake of time mainly, but also due to data constraints. So we have here an image of a community of corals. This is a photo of Ed Roberts from those submerged shoals that Tom Bridge has alluded to earlier, or in other conversations with you that I'm sure you've heard about. So if we want to quantify this among or between coral community differences in productivity, we need to be quantifying things like the growth productivity, so the overall carbon fixation of the coral community. This is kind of the energy coming into the system through photosynthesis. We then will have excretion, as I mentioned earlier, so up to 40 or 60% of the carbon that comes into a coral colony can then be excreted almost immediately into the water column, so we need to include that in these kind of systematic approaches. And then there'll also be kind of net production, which is resources which are assimilated into biomass and retained within the system. So I've broken the talk up into 2 sections to kind of deal with these processes separately. So another huge problem we have in kind of scaling up to reef scale processes is that we need to generalize among species in a systematic way. So there's about 1,500 species of Scleractinian corals. About 780 of these are reef building or symbiotic species, around 360 of them on the GBR. So whilst it's possible that we could collect data for all of these species individually, it's really not the most efficient or effective use of our resources. A sensible way to generalise across species will allow us to progress further forward more quickly in answering these kinds of questions. As an aside, I'd also just like to mention that it's actually a really exciting time in coral taxonomy at the moment. So these words exciting and taxonomy don't often go in the same sentence together, but it really is true in the coral world. So there's been a huge molecular revolution in coral systematics. There's been a redescription of the various families and genera. Everything's being repackaged, and other new species being described. It's kind of a nightmare for those of us who had to teach coral ID to undergraduate students. But other than that, it's an exciting time in allowing us to do things like map coral traits onto their evolutionary history to understand the evolution of different traits. For this work, though, stepping aside from these issues of the taxonomic revolution, there's 2 main ways we could generalise these physiological productivity processes among species. So one is based on colony morphology. Here we know that colony morphology, whether you're a mound shape or a branching, has important implications for things like withstanding cyclone damage. And another way to generalise across species can be based on polyp size. So the data that I'll show you in the subsequent slide is trying to generalise metabolic rates across species with different polyp sizes. From a physiological perspective, I think things like polyp size are actually really important because it can constrain things like food consumption from the water column. It's also true that colonies with the same morphology can have very different polyp sizes, which is what I've illustrated in the images that you're showing here. So polyp size varying from about 2 millimetres up to about 3 centimetres in these species that you're looking at. Okay, so these are kind of the first pass of data that I've been working on collecting for about 2 years now. It's actually a massive amount of effort goes into collecting just a single data point on this graph. Any of you who've measured metabolic rates of organisms will know that the challenges involved with those kind of techniques. What you're looking at here is polyp size on the x-axis, and then metabolic rate as respiration rate of those colonies on the y-axis. Each data point is a different species, and the species represented are covering a range of different morphologies, from folios, plates, to branching and then mound-shaped morphologies. And there's a fair bit of variation among species, as you would expect, but we do see a clear signal of scaling of these metabolic rates with polyp size. So the physiologists in the room will be kind of familiar with this notion of these allometric nonlinear scaling or power law scaling relationships between size and metabolic rate. In nature, we see kind of ubiquitous 1/3 and 1/4 power scaling relationships between size and metabolic rate. So it's interesting that we see about an 0.36 or 1/3 scaling between metabolic rate and polyp diameter. That's a topic of another talk that I could probably speak to you about for hours on end, but not today. So the next part of the puzzle, moving on from a way of quantifying among species variation in their productivity, is to quantify among species variation in how much of that energy is retained within the system as biomass and how much is retained as skeleton, how much is then lost from the system through reproduction. So what you're looking at here is an ordination of around 20 different coral species based on their relative energy allocation to reproduction, tissue biomass, and skeleton. So for this graph, I've assembled data from the literature together with data that I've collected myself on things like egg size, egg number, tissue composition, biomass, colony morphology, a range of different other metrics. And it's all come into this single ordination of species based on these energy allocation patterns. So this species illustrated in red here has about 30% allocation to tissue, 10% to skeleton, and the rest going to reproduction. So people who are familiar with corals and coral geometries will probably be able to tell that this is a branching species. I'll just bring these ones up. We also see in this ordination a clustering of species based on their morphology. So the massive species clustering together, obviously, because they have this much higher production of skeleton compared to tissue. So where I'm going with this is not just a tool to translate things like energy acquisition into the amount of biomass that's retained within the ecosystem, but also as a kind of functional classification or a life history classification for coral species. Okay, so the title of the talk was Variation in Community Productivity of Coral Communities with Climate Change or Global Warming. So what I've done now is taken the results from the first information of the relationship between metabolic rates and polyp size, the subsequent results showing how that energy is allocated between tissue, skeleton, and reproduction, and then converted that into community-level fluxes of carbon. So we have our two communities here that are quite different in their coral composition. You can see one which is dominated by acropora branching corals, and the other which is more dominated by those really fleshy, mound-shaped species. Okay, so in these calculations that I'm about to show you, we have the same number of corals within each community. Each of the corals are the same size, but it's just the composition of the species that's differing. So in the case of the acropora-dominated community, so the numbers here are in units of grams of carbon fixed per patch reef per hour, but don't worry about the units for now, just focus on the numbers. So we have about 8.2 units coming into this community, about 3.3 being exported as excretion, 2.3 as eggs, and then most of the energy that's retained in the system is retained as tissue biomass and not skeleton. On the other hand, for the community that's dominated by those fleshy mound-shaped species, we have less energy coming in. We have much less being released through reproduction, so less export of carbon to the pelagic environment through this reproductive process. Less energy retained as tissue, but a much higher component of energy being retained in the ecosystem as a skeleton, simply because those mound-shaped species are producing much more calcium carbonate. So summarising all this, we might expect that these acropora or branching dominated communities are exporting about 2% more carbon to pelagic environment. They have about 1.8 times more standing biomass, but around 4.5 times less skeleton. So I mentioned that point 2 in answering this question of how does productivity vary with climate change and global warming is that we need to be able to incorporate spatial and temporal variation into our calculations of productivity rates of corals. So Andy Hoey showed a similar graph earlier today showing variation along this GBR gradient in temperature. You're seeing here data from available online from this Terra Modus satellite data, where we see a gradient of about 29 down to 18 in Lord Howe Island during the spring, and then in December from 29 down to about 25 degrees. So the way to incorporate this spatial variation and also within space through seasonal variation in temperature is by using what we call a thermal performance curve. So Morgan showed one of this last night in his example for coral trout. We have temperature on the x-axis and then some measure of performance of the organism on the y-axis. In nature, we see this typical kind of hump-shaped relationship between temperature and performance. It's not always perfectly symmetrical. It's often skewed to one direction or the other, but in general, a non-linear hump-shaped relationship between the two. In this example, which is calcification for 2 different coral species, you can see that over a temperature range of 25 to 29 degrees, we see about a 3-fold decline in calcification. This kind of functional response data for corals is extremely sparse in the literature. We've done a good job in coral physiology and ecology of measuring responses at 2 or 3 temperatures, but the data to actually calibrate these functional responses is extremely sparse. So what I'm showing you here is one example of a paper we published in Global Change Biology earlier this year. It's for a Mediterranean coral species, and in this study we were attempting to measure or to detect a signal of acclimation to different temperatures along a latitudinal gradient among these 4 different populations. So we went and sampled these corals from 4 locations, 2 on the coast of Spain, 1 in Italy, and then 1 down on the Israeli coast in the Mediterranean. Brought them back to a common environment and then measured their physiological responses to temperature between about 18 and 32 degrees. You'll see these hump-shaped curves, like similar to the example I showed you earlier. And in the red stars in these plots are showing you the thermal optima, so the temperature at which these physiological processes were maximized. The green lines are showing you the environmental temperature. So if acclimation or adjustment of physiology to match temperature was going on in these environments, we'd expect to see the red stars lining up with the green lines. But in fact, in these examples, we found practically no evidence that these species were able to acclimate their physiology along this temperature gradient. It doesn't look like much in this figure, but it was going from the lowest was at about 24.5, and the highest was down to about around 28 degrees, so a 4-degree gradient of temperature change. So I mentioned that there's extremely sparse data for corals, and what I would urge us to do over the coming years is to build in more of a macro-physiological perspective to the work that we're doing, which means going out to different environments, ranging from Japan down to the southern GBR and Lord Howe, across the Pacific, as many sites as we can get to quantify this among population variability in physiology, and then to bring in genetic and molecular techniques to tease apart how much of that is genetically driven and how much of it is physiological acclimation. So that's my 2 cents for the direction I think I would like to be going in collaboration with others in the centre over the coming 7 years or so. So I mentioned that these data on thermal performance are sparse for corals. So Bob Pressey gave us a data-free image in his talk yesterday. I'm giving you a data-sparse figure in this case. So we have here examples of a simulated relationship between a physiological proxy, say net photosynthesis in this case, with temperature on the x-axis. So the green bars show you the temperature at which I currently have data for these 4 different species. I have data for more species, obviously, but they couldn't fit everything on this one figure. The error bars, or the grey lines in this case, are giving you a 95% confidence interval that I just put on to show the variability among colonies within species as well. So what I'm getting at here as an illustration of a research direction is quantifying these thermal performance curves, accounting for within species variability, building this into these models so that we can predict things like the productivity of different coral communities, given changes in community composition that we know are likely under climate change scenarios, and also incorporating this variation purely due to temperature changes as well. So take-home messages from today. The first kind of exciting finding that, well, I'm particularly excited about and I think is relevant for studies of physiological ecology in general. So we are seeing this signal of metabolic rates scaling allometrically with polyp size for corals. It's a ubiquitous relationship we've seen for all kinds of animals. Things like heart rate, organ size scale allometrically with body size in a range of different animals as well. So interesting that we're seeing this kind of 1/3 power scaling relationship between metabolism and polyp size in corals. So hopefully I've convinced you as well that we can kind of ordinate different coral species based on their physiological or their allocation of energy between reproduction, tissue and skeletal growth. This kind of process allows us to translate things like photosynthesis rate and carbon fixation into processes such as how much of that energy is retained within the system versus how much is lost from the system. It also helps us move towards something of a functional classification of coral species. So it's certainly not the case that corals are all slimy rocks. I know our fish ecologist colleagues would like to have us believe that corals are slimy rocks that are all that perform an equal function in the environment. I don't think that's the case, obviously based on these equations that I've showed you today. Another point I wanted to make is that the evidence that we have from these functional responses is that it seems corals have limited capacity for thermal acclimatization. So there's lots more to do in this space. I know other researchers around the world are using translocation experiments to measure this capacity for thermal acclimatization. It's also true that the example I showed you today was in a species that wasn't able to adjust its symbiont type. So the symbiont type within those Mediterranean corals from all of those locations was the same type. So suggesting that in the absence of the capacity to change symbiont types, we're seeing this limited capacity for thermal acclimatization. So I'd just like to acknowledge the following collaborators and also research assistants and funding bodies, and thank you for your attention.